Okay, good morning. It's 11 o'clock, so let's have a class. We're trying to finish the slides 2, and it's actual lecture 4, and then keep posting the recording on the eCampus so you can study afterwards. So we try to finish these slides, and then we move on to the new one. Again, at the beginning of each section, I will do a review of what we talked last lecture. So we talk about the last lecture, we first talk about the microscope. Since we practiced the light field microscope, so we mentioned some other microscope we has not been practiced. So we mentioned about dark field microscope. This is used for testing a special bacteria. We call it a spiral sheet. Now, why is the reason we'll have a dark field? The reason is we had a block underneath the condenser, so the light going through is perpendicular. Okay? And the spiral sheet, we mentioned about 3 different types of the spiral sheet, which is a Treponemian, Palladian, which is for syphilis, Borrelia, Burgdorferi, which is for Lyme disease, and Leptospiro interrogans, which is for jaundice related to the liver disease. Then we mentioned about the phase contrast microscope. This is basically we had a phase ring, and this phase ring will change the speed of the wavelengths. It's either 1/4 ahead or backwards. So the wavelength of the light will be changed, and it's showing the image. And this is usually for testing the internal structure of eukaryotes. Some of the eukaryotes, and we cannot do the staining, using a basic stain to do. Then we mention about the fluorescence microscope. And we also mention about the laser confocal microscope. And basically these things, there is one thing is the same. You have to do a staining. So we using a Davy stain to stain DNA. So you see a different color. You see the green color is usually a live cell, and you see the green color, the red color, usually it's a dead cell. So green color is a live cell, and the red color is a dead cell. And these two have a very good resolution compared to the dark field phase contrast because of their energy resource is different. Laser confocal is from the argon laser. Fluorescence we using UV light or like blue light. So these are something we mentioned. And then we talk about the staining technology. We very briefly mention about simple staining. And we practice in the lab. We spending quite a lot of time talk about the gram stain. You get to practice today in the lab. And we also mention about the acid fast stain. And the acid fast stain is very special, we mentioned. The reason is, it's basically for bacteria, it's called mycobacteria. And these bacteria have a very waxy cell wall because mycolic acid is very high amount. It's unsaturated fatty acids looks like a waxy cell wall. So we need using a very special technology called heat steaming with a special dye called carbon fusion. and contrast them with methylene blue, then we can see the bacteria. We can make the determination as a presumptive TB. So these are the things we have been talked already from last week, before the Labor Day. So today we're going to continue to talk about several special staining technology and also move to the electron microscope. We have left something about the staining technology has not mentioned is the following couple of them. We're going to mention about endospore stain. So endospore stain. Endospore stain has one step, is very similar to the acid prostate stain. So first of all, we want to mention what is endospore. Bacteria has an endospore. It's buried very much in the center, or sometimes in the terminal or subterminal. So it's for bacteria to protect in the harsh environments or stressing environments to survive. So why we do the endospore? There are two reasons. Number one is we want to see the presence, absence of endospore. Is there endospore? That's the first thing. The second thing is the location of the endospore. Because an endospore could be right in the center. In the central. It could be at the end, we call terminal. It also could be between center and terminal, we call subterminal. And the location of the endospore is genetic dependent. So for example, cross-treating. We're spending quite a lot of time later on talk about cross-treating in the exam too. The cross-treating, they are terminal. Bacillus subtilis or Bacillus cereus, the species we're going to use on Thursday for endospore stain in the lab, it's subterminal. And there are some other species in central. So once you know, we had an endospore there, and we know the location, we couldn't make a guess what type of the pathogen it is. Because majority of the species or strain or species or strains has endospore, the category could be cross-treated. Most likely, I just put an SPP there, or it could be Bacillus. Those are the two major common ones what we did in the lab. Okay, so that's the background for the endospore stain. The structure of the endospore we'll talk about on Thursday because we have a section for that. Now, endospore stain, it's a very rigid or very tough structure because it's resistant for the harsh environments. So what we do, the air dry heat fixing are the same. The difference is on the glass slides, you have a bacteria there. You have a circle there. The smear is already prepared. You have to flood it. Cover the whole surface with a special dye called malachite green. That's a special dye. And at the same time, we also have to use heat steaming. The heat steaming has two methods to do. You either use Bunsen burner on the top, or you put the whole thing in a water bath. The water bath set at 60 degrees Celsius. Let the heat go in from the bottom. The Bunsen burner is on the top, the heat is from the top, and then the water bath, the heat from the bottom. So you flood it with malachite green, then you either put on the water bath or using a Bunsen burner back and forth on the surface. Let it do a very intensive heat steaming for 5 minutes. Let the dye go inside. And then how we do it, we rinse and we contrast them with saffron. So once we do that, you can see the endospore. That's what it looks like. So picture B is indole. Look at those pink color. That's saffron. I can say those are trash. We don't care. What we care about is those green ones. Those green ones are indole spore. But why it's green? Because we use a dye called malachite green. That's why it showed you the green color. So this is the indole spore stain, and we will practice in the lab on Thursday. The next two we're not going to do, but I'm going to introduce to you capsule stain. Bacteria have a capsule. It's a polysaccharide. It's for the bacteria to survive in the host and cause problem, cause pathogenic. We mentioned earlier, very easy example for capsule stain is a bacteria Streptococcus pneumoniae. You see the diplococc cell have a very heavy package there. That package is capsule. So, capsule stain, there are two methods we can do. Number one is negative stain. For example, you can use Indian ink or use nigrosin to stain the background. And the capsule will show that. That is what type of the one on the picture A, it's negative stain. So you see the capsule here, which is greenish black. And at the center you see the diplococc cell, which is Streptococcus. And the background looks like purple color because you put the dye there. The nigrosin looks like that. Negative stain. The second one, sometimes we could using sulfide, copper sulfide, to directly to stain the capsule. But this is not used too often, so less likely we use. Okay, so that's a capsule stain. Next one, flagella stain. First of all, you need to know the flagella, what it looks like. Flagella composed by a protein called flagellin. So it's a little different in bacteria and fungi. In a bacteria, it looks like there's a tail. And we will talk about later, this is called monotrichos. Because of this, they have a tail, so they can do moving. And this moving is a real motility. And it also could be on the side, like this. That's called amphitrichos. Or it could be everything on one side, called lophotrichos. Or could be the bacteria covered the whole thing. That's called petrotrichos. The tricky thing for do the flagella stain, because it's protein-based flagellin, so it's difficult to using the special, like a basic dye to do it, because it's you do heat fixing, it will be melted. So that's why flagella stain is a little special. We need using two glass slides. The one is put horizontally. Another one which you put right here. Little perpendicular, but the angle is about like 75 degrees. And then you put the flagella there. You roll it very gentle. Let it roll. Because it's melted usually. You roll it like that. And then, like you did in your biology lab for the bloodstream blood cell, you're very gentle to using this glass slice to move. Okay, then will become like this. Then it covers the whole surface. Then we have to do using formalin to fix it. To fixing it. And then we're going to be adding silver nitride to do something we call a coating. Then you can see the flagella stain, what it looks like. It looks like the picture like that. It's a flagelling stain, actually. You're staining the flagellum. You also could also using carbon sulfate to directly staining the tail, the flagellum, but barely use it. Okay, so there are two methods we can use. Now for the fungi, it is different. Fungi, what it looks like. The center you have 2, and outside you have 1, 2, 3, 4, 5, 6, 7, 8, 9. So fungi, we call it a 9 plus 2 structure. That flagella looks like this. Okay, so that's a little bit different from the bacteria and also the fungi. We're not going to do capsule stain and the flagella stain in the lab. Just give you some of the introduction. This gives you some of the examples. The staining we already talked. We talked about the gram stain. We had a very good example today because I bring the species called Enterococcus facilis in my lab compared to the E. coli, what you have in the teaching lab. The E. coli looks like it's a little bit contaminated. So you will see the E. facilis looks very good for gram stain, and the E. coli you may find here and there mixed with gram positive and gram negative. It's interesting to give you impression of what the cross-contamination looks like. So that's the first one, is gram stain. This is the acid-fast stain we already mentioned about using carbon fusion. And the endospore stain for bacillus looks like that. Capsule stain. And the last one is flagella. This is a bacteria we will mention sometimes in the lab called Proteus vulgarius. The words is very small, called Proteus vulgarius. This is a bacteria, is belonging to Petrotrichus. The flagella is completely covered the surface, which means it has a very strong moving capability. How strong it is? This bacteria will not forming single colony on the agar plates. On the agar plates, what it looks like? Looks like this. They are completely a cluster, and then you pick from there. So this is kind of thing we call swarm. And the Proteus vulgarius is a very good one for biochemistry test, because all kinds of the biochemistry test, sulfide motility test, phenylalanine deaminase test, nitrite reducing test, and 3-tube fermentation test, it's all positive. So it's a very good lab material to showing you what the positive of the biochemistry results looks like. Now what it looks like here is looks like the picture. You can see it looks like a worm. Everything is there and it's moving. Can feeling about like a bug, like a big bug is moving on the agar. Because it's called a Petrotrichus, it's flagella, completely covers the surface. Okay. So we finished talking about these staining technology. So if we add up here, we have endospore stain. Then we talked briefly about the capsule stain. And we also mentioned about the flagella stain. In the lab, we did simple stain, gram stain, we'll do endospore stain. Acid-fast stain, I used to have mycobacteria because of the preservation is not very good, and also the pandemic. We stopped the lab for about a semester. After that, it's dead. So we no longer can do the acid-fast stain. Otherwise, it's pretty much fun. We can do that. So these 3 we're not going to do. We're only going to pick 3 of that to practice in the lab. Okay. Okay. Next one. we're going to talk about electron microscope. Electron microscope obviously has a very strong resolution capability, and we can using that to do the observation of very tiny microorganisms. For example, virus, and we already mentioned. So we want to talk about the principle behind it. A little detail, not too much. An electron microscope basically has two categories. One we call scanning electron microscope, or acronym we call it SEM. SEM basically is the surface. That's why we call it scanning. It will give you a very similar but more clear 3D images compared to laser confocal microscope. So the resolution and the magnification is not too huge. Somehow like 5,000 to 10,000. Typically 7,000 to 8,000 magnification. Not too much because we want to see the outside. Another microscope is called means transmission electron microscope. That's called AEM. Transmission electron microscope. We can see clearly the internal structure. So the magnification is huge. Somehow 10,000 could be up to 100,000 magnification. It's huge. Now, how about our light field microscope? Remember, we talked about in the first class, light field microscope is only 1,000 with oil immersion. Is that right? So I should add a plus there. Cross there. Good. So let's talk a little bit about the electron microscope. What's the principle behind it? You have a picture there. Let's do a comparison we can do. So this is a light field microscope. This is the electron microscope. So what's the difference? I'll draw from the bottom. Okay. Light field microscope. Let's say we have something like this. We talk about in the lab. Okay. You have a stage. Then you have a condenser, then you have a light like this. So first of all, the energy resource obviously is light. It's general light bulb. Then you plug it in, you turn it on, the light is coming. Then you go in through the condenser. Okay, you go here. Some go here, some will be reflected. Okay. Something like that. So light, and you're passing through. This is we call objective lens, and here you obviously have something you can see. Use your eye. We call it the ocular lens. And this is also glass. So when the light passing through, what you see is air, is basically glass, and this is also glass, and this is your naked eye. To do the observation. That's what light microscope. Okay, what is the electron microscope? instead of we have a light as an energy resource, I have electron beam. Instead of we have a glass as an objective lens, as a condenser here, this is a condenser. This is also glass, okay, for light microscope. The condenser here, what it looks like? It's like a donut shape. So we call donuts condenser. It's made by electron magnet. Okay. And then you don't have this like a stage. You go up, you have an objective lens. This is also donut shape, which is also electron magnets. And then you go up the ocular lens. The ocular lens is also a donut shape, is an electron magnet. And then on the top, instead of you use naked eye to do the observation, this will be a big viewing screen connected to the computer system. And instead of you have a light passing through, this is basically let's say you go here. These electron, we call it an electron air. That's a little difference between them. This is air, this is electron air. Or we say electron rays. This is light, this is electron rays. Another big difference. This is the whole thing is open air in the light field microscope. For electron microscope, you have to in a vacuum system. What means a vacuum? Basically, which means no air or no oxygen. The reason is the electron rays could collide with air molecule. It will bother the image formation. So, transmission electron microscope, you see a big tunnel there sometimes. Look like a high tunnel facility to planting the tomato at West Virginia. Very similar, a huge tunnel there. A whole tube, it's in a vacuum system. And how do we know it is become a dark? How do you know the images become a light? It depends on the density. So we call it the electron density. Okay, if you see it's a heavy density area, it's usually become a darker. You see the light density, it's usually bright. So that's the story of the whole structure, the difference. Here we have air, glass. Here you have electron rays. Here you have a light. Here you have electron beams. Now how are we going to make the sample? We're using electron microscope. We need to get a training, a very strict training. Sometimes need a certification program to operate it. At the shared facility at WVU in the engineering building, the building behind our teaching lab, the South Agriculture building, they have some training program for that. But for bacteria, it's not allowed to use pathogen. It's only for surrogate bacteria. So basically the sample, you have to be, first of all, is do a liquid nitrogen freezing. That's the first step. The second step, you have to be slice it very thin. Because a thin slice will let it be concentrated with the electron rays instead of collide with heavy air-traveled molecules. Then, after we slice it, you will see the area looks like this. So we call this is like a shadowing place. And this shadowing place, we have to do a heavy metal coating. And basically we use lead. Sometimes also use silver nitrate, we use lead to do the heavy metal coating. And after that, it took about a couple of days to prepare, so we can use the electron microscope to see the internal structure. So the processing is different. That's why the electron microscope, when we're using, we not say staining, we just say coating. Its terminology is a little bit different. Here I give you a picture which is showed about the transmission electron microscope and the scanning electron microscope, just to let you know the basic difference. And you using AEM, you can see the ultra structure of these thin sections. For example, you can see the ribosome, you can see the cell wall, you also can see the nucleoside. I would say this picture is pretty old. It's a white and black picture. Right now we can all get a color picture. We can get a 3D images. And this is using scanning electron microscope. You can get a 3D images, very beautiful. And from the outside you see the whole situation, what it looks like. It's typically very good for parasites. Let's say Toxoplasma gondii, you have these pictures, you see very clearly what it looks like. And some research even can see how they penetrate into the host cell. It's like a movie, a cartoon movie. It's really beautiful. The only thing is it's expensive, and unfortunately we cannot practice that in the lab. Usually those facilities, somehow like $200,000 to $300,000, we need to apply a special grant from, that's called equipment grant from USDA or from NSF. Then you can set up there. But the big problem is, any of those facilities, if they're cracking down, or there anything problem there, we call it troubleshooting. We need another service charge for that. So that's why like the university like WVU, we only have 1 or 2 facilities there. Okay. So last 2 slides. We mentioned about these already in the lab. We just want to emphasize one more time. This is a picture showing you why we need to do the staining. We back to the bacteria because we can see the shape and the arrangement. Shape, we usually give them a terminology. Let's say cell shape. We give them a terminology called morphology. So what means morphology, we mentioned a couple of times, is called a cell shape. Arrangement, which means how they group together, you can understand it. So here you can see coccell. It's a sphere, it's like a ball. It could be diplococcell. It's a pairs, like Streptococcus pneumoniae. It could be Streptococcus. You will see today we are using Enterococcus faecium to do the gram stain. It's a chain. Because Enterococcus faecium a long time ago belonging to Streptococcus. Staphylococcus, you observed the use you made the glass slides in the first lab. That's Staphylococcus aureus. Looks like that. Tetrade. Micrococcus ladeus should be that. We don't have that species anymore. It's 4 of them stacked together because of tetrade. Sacini, which means there are 8 coccels stacked together. This is a very interesting one. The picture showed here called Streptococcus agalactiae. This is the one is a sexual transmitted pathogen, typically for sexual activity women around 28 years old. We have a nickname for that. We call it honeymoon bacteria. We'll mention this again at exam 2. Just to give you some hints right now. Some others for the morphology and arrangement. Bacilli. That means lots. You already see E. coli. Some of you get a good E. coli slides, which means it's short lots. Some of your slides is actually not E. coli. I think it's bacillus because it's large. But lots of the pathogen is coccobacillus. We will mention in the exam too, a lot is coccobacillus. But vibrio, it's like a comma shape, looks like that. The picture A, which is showed you, is a vibrio. Vibrio coral, that is a major pathogen for the seafood, like jellyfish. The problem for this pathogen, it will be causing you dehydrate. You will lose 5 liters or 10 liters of the water in like an hour. So the treatment for vibrio coral is not use antibiotics, is rehydration first. You need to put the water back. So that's a vibrio. Sphaeria, something like a special bacteria called Campylobacter jejuni. This we will talk later. Campylobacter. That's called Campylobacter jejuni. This is a major pathogen for poultry meat. Same category with salmonella. This is a very ironic pathogen because it's microallophilic, which means they grow in the environment, require very limited amount of oxygen, only 5 to 7%. So it's very difficult to grow in the lab, but it's a big concern for the poultry industry. That's a Campylobacter jejuni. We will talk later on. In our last section when we talk about applied microbiology. Spiral sheets we already mentioned. Using dark field microscope, you can do the observation. Last one. This here, Leptospira interrogans, look like a hook shape. It's a very typical one for spiral sheets. Besides Treponema pallidum and the Borrelia burgdorferi. So I think these 2 pictures are very important because they are connected with exam 2 when we're going to talk about the pathogen. You can go over to study, back to study it again when we move to that. Because they give you a conclusion of the common bacterial morphology and arrangement we're going to talk in the class. Okay, so here, we finish the section of our slide 2. And we're going to move on to our next section of lecture 3 slides. Lecture 3, we're not going to finish it today because it's have a lot of material being covered. We're going to mention some of them today, some of them on Thursday. Something I will be emphasized, it is important. So we need to moving all these down. So we talk about the microorganism. We talk about history. We talk about how to isolation bacteria. Then we talk about staining technology and the microscope. So the idea is using these tools, we want to see the content, or we say the internal structure of a bacterial cell. Some of them are very similar to the one you learn in your biology class, but some of them are not. So we will be talk about here. First question is very simple. You have 2 pictures here. A, left and right. Prokaryotes and eukaryotes. What's the major difference? A couple of years ago, I have this question in the exam. Some people just say, prokaryotes is simple and eukaryotes is complicated. Not really. Although you see prokaryotes only have a couple of named or labeled structure, does not mean it's simpler than eukaryotes. Because think about the bacteria and the fungi, they have to survive in a various environment. They have to be still do the same thing. So, although it's simple, but it's more functional, which means maybe more complicated, or something we don't know yet. Now, what's the major difference? Said very simple. Cell boundary structure. Is that right? You have a cell membrane system to separate it. Eukaryotes has it, prokaryotes doesn't have it. A very good example for microorganism. Bacteria looks like on your left side. Fungi, molds and yeast is on your right side. Now, what are the structure are similar? They all have ribosome. They all have cytoplasm. They all have cell membrane. They all have DNA. But be careful. Cell wall may be the major difference. Bacteria has a cell wall. Plant, animal cells, even fungi, they do not have a cell wall. And you already learned the eukaryote structure in your biology class. So, we're not going to go over the right side picture or spending 45 minutes to talk about that, because that's a biology class, or the animal physiology class, or plant physiology, plant science class, they already talk about that. We're going to focus on the left side for something really special. So we go down here, the prokaryote cell structure. It's more clear, something we're going to focus to talk about. A bacterial cell, you can see, very special. You have a capsule we just mentioned, cytoplasm, ribosome, cell wall, plasma membrane, nuclear area, DNA. That's what we're going to talk in the exam 3, the DNA replication, transcription, translation, but focus on the bacterial plasmid, capsule, fimbrial, and the flagella. We just talked about the flagella staining. So those are the structure we will mention. And it's very nice. We gave you an overview slides. What's the function of these? So cell membrane, it's a barrier, boundary, transport of the nutrients. Metabolic process, the signal detection. Cell wall give you shape, protection. Ribosome, both eukaryotes, prokaryotes have, it's a protein synthesis. The detail we'll talk about in the exam 3. Capsule, we just mentioned, the resist to phagocytosis. Villi, it's for the minor motility. But more important is for the DNA, RNA, or genetic information transfer. So we call it conjugation or transformation. Flagella, true motility storage, granule is inclusions. We will be mentioning storage granule in our exam 2. The first pathogen, we talk about cornobacteria diphtheria. You will see a granule there, which is for storage of phosphate. So this slide is important to give you an overview of prokaryote cell structure. So we want to mention several things. Okay, so first one. This sounds easy. Everybody knows, but we want to talk about this. Everybody knows our cell membrane system is composed by two major components. Yes, a dot with a tail, that's called lipids. Or sometimes we call it a phospholipids. So we're going to draw a couple of them. Okay, one chemical there, usually it's not stable. So we need another one. What is the other one? It is interactive. That's protein. Protein has a different protein. Some of them across the membrane we call transmembrane protein. transmembrane protein, lots of the time, like a channel for transport material outside, inside. Nutrients go inside, toxication, metabolic results come out, or waste come out. It could be something like this. It also could be something like that. This is we call peripheral protein. Now sometimes you also going to see something like this. That's sugar because of oligosaccharides. It's a minor component. But the major components for cell membrane is lipids. We call phospholipids plus proteins. And they are in a balanced moving status. We call it fluid mosaic model. Now we want to talk about these. This guy. This phospholipids. Here, hydrophobic. Here, hydrophilic. Is that right? No. Opposite. Then when you're doing the exam, you just guess. Sometimes you guess right, sometimes you guess not correct. I'll tell you why. Okay. Hydrophilic head and the hydrophobic tail. What means hydrophilic? It's like water. What means hydrophobic? Dislike water. Or we say cannot dislike water. Okay. You sometimes remember, sometimes doesn't remember, or mess it up. Sometimes you have to guess. But I tell you, if you know the chemical, you will be chemistry, you will be remembered. This one here looks like a slides, is an intro level. So we want everybody to be good, to well equipped for knowledge. So we want to make it complicated. So we're going to draw the structure of that. What it looks like. At the very beginning, it looks like this. Then you go here. So you go here. You go here. Then you go here. CH2, 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 CH3, CH2, 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 CH3. Why are we going to draw this? Here, on the top, called ethanolamine. What means ethanolamine? It's look like an ethanol. Ethanol is another terminology for alcohol. An alcohol dissolved in the water. So the head is hydrophilic. So you remember that in the head, looks like there's something like alcohol, or alcohol modified products called ethanolamine. Ethanolamine. So you will never forget about it. This guy, you know, phosphate. We talked about it already. How about here? Glycerol. How about here? Lipids. Is that right? Does lipids dissolve in the water? Of course not. You buy, put 1 drop of the oil, your vegetable oil, put on the water, you make a soup, it's floated. Like I always ask people, what is the definition for lipids? Is some chemical will be dissolved in the organic solvents, not dissolving in pure water. That's lipids. And you may don't understand about here, but if I put it this way, you will be more familiar with it. Is that right? We go here. The triglycerols. Same thing. That's lipids. So then, if you know C16-0, what is that? 16 carbon, 0 double bond, saturated fatty acids. What is that? Stearic acid. Or palmitic acid, sorry. That one we don't like it. Okay, C18-0, what's that? Stearic acids. In beef, lots of stearic acids. We don't like that one too. We like this guy. What is this? Linoleic acid. Okay, what's the other one? Linolenic acid. Is that right? Are these dissolved in the water? Of course not. So you'll remember that. In other words, when you see, this is the alcohol, modified products on the top, this is lipids. That's why hydrophobic, we all call it lipophilic. In other words, another name called lipophilic. So that's very simple to understand the chemical structure, then you will remember it. The hydrophilic tail and the hydrophilic head and the hydrophobic tail. And it is interacted with protein, and the protein is also always there and for the moving, in a moving status. And this slide showing there is very basic. Okay, we get a little bit deep there. Cell membrane system. That everybody knows. It could be the cell wall synthesis. It could be energy metabolic. We're going to talk about DNA replication, sense of the stimulus, the molecular transport. It's a function. That's a conclusion slides, but we want to talk about detail. These looks like everybody knows, but something we want to emphasize. So we want to talk. Uptake of the nutrition for bacterial cell. You already learned in your biology class how human being or animal cell to uptake the nutrients. And pretty much in your high school you know about that. Bacteria keep some of that, but some of them are new. Passive diffusion. This is everybody knows. The requirements from high concentration area to low concentration area. No energy required, no ATPs, and no carrier. Which means permease, it's an enzyme, no carrier. Does bacteria use passive diffusion to get the nutrients? Yes, but only water, oxygen, or some of other small chemicals, carbon dioxide like. So passive diffusion they do use. Second one, little difference between passive diffusion, we call it facilitated diffusion. What's the difference between that? Still high to low concentration, no ATP required, but needs carrier. And you also learned about in your biochemistry class, this reaction curve is very similar to that. X-axis is a reaction rate, y-axis is a concentration, or x-axis is a concentration, and the y-axis is a reaction rate. It will become flat in the end, because the carrier is permeance. It's an enzyme. The enzyme, based on Michaelis equation, it will become saturated at the end. So that's called a facilitated diffusion. Does bacteria use it? Yes, but very few. Why? Because in a bacteria's living environment, especially without the liquid media, it's always the poor nutrition outside of their cell wall cell membrane system compared to the liquid media. So for example, bacteria on the bench. It's always poor nutrition. We don't put any sugar there, we didn't put any protein, put any lipids there, but they can survive. Certainly they're using other methods. So facilitated diffusion barely used by bacteria. Most of the time used by eukaryotes. They could engulf, they can get it. Well, that's facilitated diffusion. This one everybody knows, active transport. Active transport, lower to high concentration, needs energy, needs ATP, needs carrier. Does the bacteria use it? Of course. Some of the sugar, some of the carbohydrates, they need ATP transport, active transport. And we always call it's called using by ATP, so we call it an ATP binding cassette. Is that right? You learned in your biochemistry class. That's called the ATP binding cassette. So we call it an ABC system. Okay, this is you already learned. But I want to tell you, the bacteria nutrients transport using passive diffusion, facilitated diffusion, and active transport, about like 40%. 60% of the time, they're using group translocation. It looks like this picture. Now you're going to understand about this one, you have to understand glycolysis. So biochemistry, you want to talk about this. So what happened? You should have learned this in your biology 101, or some of you taking a biochemistry class right now, then you get very familiar. We have a glucose. The glucose like this. Okay, that's a glucose. We'll get to these things later on. That's a glucose. Is that right? Glucose have a process. Looks like this. Will generate pyruvate. And this name we call glycolysis. So we can go real quick. The glucose becomes glucose 6-phosphates, then become fructose 6-phosphates, become fructose 1,6,5-phosphates. It's right here. There you have a chain. So the chain will have, let's say this chain we move here, glycerol behind 3-phosphates, 1,3,5-phosphoglycerate. Then we go 3 phosphoglycerate. Then we go 2-phosphoglycerate. Then we go to the key point here. Phosphoenolpyruvate. The last one, pyruvate. What are the keys? Is right here and right here. More important is this guy we're going to talk. What is this? Pyruvate. What is this guy? Phosphoenolpyruvate. Okay, bacteria going through glycolysis. What's the difference? You learned in your biochemistry class. Glucose becomes glucose 6-phosphate. Hexokinase. Is that correct? Yes. It's only for animal and human cell. That's why you learn your biochemistry class. Here, it's not. They rely on this guy. This guy have to be recycled to go here. Then this will work. But the PEP, phosphoenolpyruvate, is not work independently. They work by a group of the other chemicals. Is right here on the picture. When the PEP becomes pyruvate, when this guy from here, the phosphate is transferring. The phosphate is transferring to enzyme 1. The phosphate further transfer to the heat-stable protein. It's HPR. And then they go to complex 1, complex 2, complex 2A and 2B. And then they help the sugar goes in. What are the sugar goes in? Glucose and mannitol. Glucose directly becomes glucose 6-phosphate, and the mannitol directly becomes mannitol 1-phosphate. Why? Because bacterial cell has a very short lifetime. It's sometimes 18 old. We'll talk about the growth curve real quick next week. It's not like a fungi can stay there longer. They need to be more economic to get their energy. So they need to rely on PEP and the other group like enzyme 1, like heat-stable protein, like enzyme 2A and B complex to transfer phosphate, become glucose 6-phosphate, go into the cell membrane immediately. That's why for a bacterial cell, when they get those carbohydrates, especially like a mannitol or glucose, they're using a different mechanism, which is called a group translocation. That's why here I would love to emphasize this slide is the most important compared to what you learned. Because for bacterial cell, the other 3 mechanisms doesn't use too much. Group translocation is much more focused and much more used. The reason is from the energy standpoint is more economic and more efficient. That's the reason. But for you to understand about that, you need to know the glycolysis, at least to know the name of these chemicals. Where the PEP is right here. When they go here, they go back. The phosphate could be transferred. Because glucose becomes glucose 6-phosphate. This means more energy potential once you have a phosphate. Phosphate is a very magical stuff. So, people did a lot of research. They gave them a name called sugar phosphotransference system, which rely on phosphoenolpyruvate. Glucose, you will learn, used a lot by bacteria. Mannitol also used a lot by bacteria. For example, Staphylococcus aureus. It grows on a very special media called mannitol salt agar. They need to use mannitol to grow. How they got mannitol is going through group translocation. Mannitol can directly become mannitol 1-phosphate. Then going through modified glycolysis. What you learned glycolysis use glucose is a standard procedure. If you change to mannitol, it's a similar one, but change the chemical's name. So you can simply understand it's modified glycolysis. Okay, that's all we have here. Then Thursday, we're going to talk about cell wall, cell membrane. We'll try to finish the session.